My name is Emily, and this video will be covering genetics and inherited disease. Firstly looking at DNA and genes, then genetic conditions, and finally some ethics surrounding the topic. I hope you enjoy listening and watching. So, what are genetics? The basis of genetics starts with DNA, and DNA simply means the information needed to make up a functional living being. Our DNA is made up of sequences, which are made up of bases, and each base has a pair. A specific section of DNA is called a gene. The gene is then transcribed to code for amino acids, which then make a specific protein. Our cells have to decode in the information needed to synthesize proteins. This is called gene expression. DNA is important for protein synthesis because it is the very first step in instructing the code for proteins. It acts as a template for transcribing the information. In order for protein synthesis to occur, the DNA needs to be copied. mRNA is made in the nucleus. This is essentially a copy of the DNA. This RNA is the messenger RNA. This is produced in transcription. The sequence of the mRNA has the instructions to make a specific amino acid. After this, the mRNA leaves the nucleus and goes to the ribosomes to be transcribed. The ribosomes then reads the codons in the mRNA 3 bases at a time. The tRNA then matches the codons in and acts as the physical link between the mRNA and amino acid. This is essential for protein synthesis because the information needs to be paired exactly right to ensure the correct protein is made. This is all being paired in a long growing polypeptide chain, which will eventually make a protein. Ribosomes hold and read the mRNA, so the codons can be matched to the correct anticodon on the tRNA. The chain builds and eventually creates the polypeptide, which will then go on to make a protein. When discussing point mutations, it is important to understand that the bases in the DNA are matched in pairs. The bases in DNA are G, C, A, and T. G matches with C and A matches with T. For RNA, bases are G, C, A, and U. G matches with C and A matches with U. Each set of 3 RNA bases make an amino acid. For example, in the sequence, if CGU is red, then it will code for the amino acid arginine. Point mutations are where there has been a change in the genetic code. There are certain types of point mutations: missense mutation, a nonsense mutation, frameshift mutation, and silent mutation. And there are certain effects that can happen as a consequence of a base being altered. This can change the amino acid sequence and protein function. A missense mutation is where a base pair changes cause a change in the mRNA codon so that a different amino acid is made. For example, AAA is matched to TTT, but the change would be GAA to CTT. A nonsense mutation is where the base pair change alters the mRNA codon for a stop codon. This is either UAG, UAA, or UGA. The effects of this causes the process to stop before it was supposed to. Therefore, the polypeptide chain made from the amino acid is shorter than needed. Therefore, its function may be hindered. Frameshift mutations often result in proteins that don't function. This is where an insertion or deletion of bases causes a disruption when the codon is being read. If one base has been changed, the rest of the amino acids are going to change, which results in an incorrect protein. In this example, the U is not there, therefore the whole sequence changes. A silent mutation is where the base changes but the amino acid stays the same because there are various versions of codons. So this does not have effect on the protein being made because they don't disrupt the sequence. In this example, CUG and CUA make the amino acid leucine. The next step is to look at the inheritance of disease, and the Mendelian genetics model in relation to humans will help explain how genes are passed from parent to offspring. From this, we can look at the inheritance of cystic fibrosis and the inheritance of gender and sex-linked conditions and chromosome abnormalities and the role that they play within inherited disease. Mendel's work is considered the foundation of modern genetics. His work was based on breeding pea plants and observing the characteristics of the flowers that followed the crossbreeding over multiple generations. These are today known as phenotypes. This can be applied to human inherited disease because genes passed down from each parent may be dominant or recessive. Therefore, patterns of inheritance can be tracked because the diseases are caused by mutations in a single gene. Cystic fibrosis is an inherited autosomal recessive disorder. This means both parents need to have the faulty gene that enables cystic fibrosis to be present in a child. Each parent has one faulty gene. This means that they are carriers and do not have the disease. The likelihood that a child would have cystic fibrosis with both parents with the faulty gene is 25%, as shown on this Punnett square. Gender in humans is determined by the sex chromosomes, which are the 23rd pair of chromosomes that humans have. These are given from each parent during reproduction. Chromosomes are tightly packed DNA, which contain genes. Males have one X and one Y chromosome, and females have two X chromosomes. There are differences in the X and Y chromosomes. The X is larger than the Y chromosome, therefore, the amount of genes carried on the Y chromosome is fewer than the X. The inheritance of gender is important because it directly relates to sex-linked conditions. They are influenced by genes found mainly on the X chromosome. Because males only have one X chromosome, if there is a faulty gene on the given chromosome, then they will be affected. However, if a female is given one X chromosome, which is carrying the abnormal gene, and the one X that doesn't, they will not be affected. In this example, let's say the gene is responsible for colour blindness. The male is healthy and doesn't have a faulty gene. The woman has one faulty gene and is normal because she has another healthy gene on the other X chromosome. If a boy is produced during reproduction, they are 50% likely to have colour blindness. The male could give the Y chromosome and the female the faulty gene. The male could give a Y chromosome and the female the normal gene. However, if a girl is produced, then they are 50% likely to be a carrier or all have healthy genes. The male could give the normal X chromosome and the female the faulty chromosome. This would result in the carrier. Or, for healthy genes, both male and female give normal X chromosomes. Chromosome abnormalities can result in many inherited diseases. One of those is Down syndrome. This happens during meiosis, which is cell division during reproduction. In meiosis, the chromosomes need to divide equally. Humans have 23 pairs, as mentioned previously. This syndrome affects chromosome 21, also known as trisomy 21. During meiosis, an extra chromosome is added from either the mother or father. This means that the chromosome has not split apart properly. This is called disjunction. Some genetic conditions like Down syndrome can be tested for before a baby is born. This is an example of genetic screening. During this, a sample is collected in utero in early pregnancy, and the fetal DNA is extracted and analysed. Then a risk percentage of the condition is sent back to the parents of the baby. The basis of genetic screening for various genetic disorders is as follows. Live cells need to be taken from the subject. The DNA is then extracted from the sample and amplified using a PCR test, which copies the DNA. In the PCR machine, the DNA is heated up to break the bonds between the double strand, which means that they are separated into single strands. Then the DNA is cooled so the primers can attach to both strands. Then, depending on what the screening for the DNA can be either processed with gel electrophoresis to separate the fragments by size, or a specific primer may be used to detect point mutations. Moving on to DNA recombinant technology. This is a technology where the DNA is taken and transferred into another person's DNA. It is technology that alters genes. This is one of the ways in which the DNA is used after it has been copied. This technology applies to many diseases, such as diabetes type 1. A person with this disease cannot produce enough insulin in their body to regulate their blood sugar levels. They are missing genes that are responsible for some immune system function. The good news is that insulin has been made by using this technology, and it was the first protein to be sequenced in the 1950s, so that people with diabetes type 1 can live a normal life. This technology can have a big impact on people's lives, as just mentioned previously with diabetes. However, there are more moral and ethical issues that are present with other means of use. Copying DNA enables science to create clones. Changing DNA to cure or help a disease prolongs life. And where that DNA is stored and kept, we can ask the question, who owns the DNA? And is it ethically good to keep it stored for potential use for future research? And can treatment cause more harm than good? In the United States, there were cases where stem cells were used on patients that had tried all the other options medically. It caused harm to the patient. First of all, the patient was vulnerable, desperately wanting medical help. This technology is also expensive, and with the result not being good, this patient was financially, emotionally, and medically harmed. This shows the importance of the responsibility of medical professionals and the importance of the correct information and not taking advantage of people's vulnerable states. On the other hand, this technology can help prevent global disease. It has already helped develop lots of new vaccines. While medicine can be wonderful, it cannot help in places where poverty is heavily present and policies and laws in other countries are preventing change from happening. In this case, it could be that money can be put into research in countries that are more accessible. This poses the question of, is it right to disturb the world, or should we leave nature as it is? I hope you've enjoyed this presentation on genetics and inherited disease. Thank you for watching.